Okay, let's unpack this. We've got this source material, looks like design notes, some debugging stuff too, for a simulation of auditory hair cells.
Right?
And our mission for this deep dive is really to sift through these notes, understand how they tried to model, well, the incredibly complex hearing process.
It's not just code, is it? It's meant to be a digital version of what might be happening in your ear.
Exactly. And the notes give us that peek behind the curtain. Now, what really caught my eye, the main goal mentioned, was boosting the biological accuracy.
Yeah, making it more real.
So that meant adding things like a tonotopic map, separating inner and outer hair
Well,
cells.
building in that Cochlear amplifier effect.
Right, and showing the neural output not just as on-off, but as a proper firing rate. Let's see how the notes say they actually built that.
Well, starting with the basics, the notes show they modeled two main hair cell types. You've got the inner hair cells, the IHCs,
Okay.
which the notes frame as the main sensory cells, the detectors, basically.
Okay. And the others.
Then there are the outer hair cells, OHCs. These are positioned more as the active amplifiers in the system.
Okay, amplifiers. But how do they know where to amplify? That brings us to the tonotopic map, right? The sources mention that a lot.
Absolutely. And it's not abstract in the simulation. The notes detail how they designed it, so specific sound frequencies make the simulated basilar membrane vibrate at specific spots.
That classic pitch to place idea, high sounds here, low sounds there, just like the real Cochlear's layout.
And that spatial map is critical,
Okay, yeah.
tells the OHCs where to do their job. The notes on the Cochlear amplifier are really interesting here.
How so?
They explain that when the sound input, the amplitude is below a certain level. I think the notes mentioned tuning it to maybe 50% of max.
Okay.
Then the OHCs, right near the peak vibration point for that sound frequency, they become active.
Active meaning they actually boost the wave's movement. They amplify it.
Exactly. They amplify the displacement. The notes even give a number like an amplification factor around 1.5 or so.
Wow. So they're literally turning up the volume locally for quieter sound.
Pretty much. And the design notes mention a visual cue too, like making the active OHCs glow yellow.
Oh, that's cool. So you can actually see the amplifier working in the simulation.
Yeah, it makes it much clearer. And it's that amplified, boosted wave movement that the IHCs then detect.
Right.
The notes connect the activation level of those IHCs directly to the chance of getting a neural signal out.
And that neural signal part, it's shown as a firing rate graph, isn't it? Also in the notes.
Not just on or off. More IHC activation means a higher probability of seeing those little spikes pop up on the scrolling display.
Like simulated neuron firings, trying to mimic how real neurons respond more often to stronger inputs.
Exactly. Now, looking at the debugging notes, it wasn't all smooth sailing.
Oh.
Well,
Ah.
there's one note, it's quite detailed actually, about struggling to get that firing rate graph to show up properly on the screen.
So the calculation was working, but the visualization wasn't. Seems like it. The simulated spikes were being generated, but they just weren't appearing until they implemented a specific fix for the display part.
Huh. That really highlights that it's not just about getting the biology model right, but also figuring out how to show these complex dynamic things accurately, especially something probabilistic like firing rate.
Definitely.
So digging through these design and debugging notes, what it means for you listening is you get this insight into how they tried to visualize these amazing biological details.
Yeah, how pitch maps to place.
How your ear actively boosts quiet sounds with those OHCs.
And how the whole mechanical vibration thing gets translated into that neural code, that probabilistic firing your brain interprets.
The notes really show the thinking behind trying to build a digital copy of this biological wonder.
So we've seen how simulating these parts, the tonotopic map, the IHCOHC roles with the cochlear amplifier, that probabilistic neural firing brings it to life. The design notes map it out.
And the debugging notes remind us it's tricky to get right, especially the visualization. Which kind of leads to a final thought. The notes showed this real effort needed just to display a dynamic, probabilistic signal like neural firing accurately.
Right.
So if showing the output clearly is so crucial for understanding, how much does our current ability or inability to visualize really complex biological systems actually limit our power to model them effectively in the first place? Something to think about.